It's my pleasure to introduce to you our next first-year Next Generation leader, Renata Batista Brito. Renata did her graduate work at NYU, focusing on the role of genetic factors in interneuron development. As a postdoctoral fellow at Yale, Renata investigated how specific populations of neurons affect sensory processing and behavior. Renata will start as an assistant professor at Albert Einstein School of Medicine in the beginning of 2018. She's interested in understanding how the development of inhibition shapes sensory representation and how this process is altered in neurodevelopmental disorders. Please join me in welcoming Renata.
Thank you. It is a pleasure to be here listening about such great science, and it has been a privilege to be part of the NGL program and to interact with my colleagues and as well as everybody else at the Allen Brain Institute. So thank you. So I'm going to tell you today about a cell type, VIP cells. It's one of the interneuron types, inhibitory neurons, and I'm going to tell you how if you perturb its development, how it impacts cortical circuits and eventually cortical function. So here I'm a little bit embarrassed with this scheme now. There is a very minimalistic scheme of the cortex. And that is where I'm showing some of the very reductionist view of the main types of cells. That I would call them sort of cardinal cells. We know now, after seeing Ed's talk yesterday, that there's the group of the inhibitory neurons. There are not just 3, but could be subdivided in at least 50 types. And the excitatory neurons, here represented by 1 type, can also be subdivided. But the main cardinal types are the pervalbumin-expressing interneurons that are fast spikers and target the soma. Somatostatin-expressing interneurons that are dendritic targeting. And then we have the small subgroup of the inhibitory neurons, which are the VIP-expressing interneurons. They have been sort of neglected for many years, but over the recent years, there has been a lot of attention paid to this cell type due to the fact that eventually to the excitatory neurons, they are disinhibitory, given that their main targets are somatostatin interneurons and inhibitory neuron itself. So I wanted to actually modulate the activity of VIP interneurons during development, and I decided to do so by removing a gene called ERBB4. It's a gene, it's a receptor, it's a tyrosine kinase type of receptor, but that binds its molecular counterpart, neuregulin. And it's a gene that has been linked to schizophrenia, and therefore my interest. And also, it has been shown in pervalbumin cells, it never had been looked before in VIP interneurons, but in pervalbumin interneurons, it was known to reduce the glutamatergic input into these neurons, so you could sort of reduce the activity of VIP interneurons by deleting ERBB4, and that was the idea, right? So at the time, this was, I had a lot of pushback as why am I going to look at this cell type from the context of schizophrenia, and why do I think it is a good candidate to actually modulate broadly cortical function, given that they are about 1% of the total number of neurons in the cortex, so a minority. So I thought, and I still think, that they are very interesting because they receive local input as well as long-range input from other parts of the cortex and other parts of the brain, and therefore some of the patterns of alteration in the brain of schizophrenic patients are actually alterations in long-range projections. But they also receive not only glutamatergic inputs, but as well as acetylcholine and serotonin inputs. And so therefore, I thought that they are in a really good place to sort of shape a broader cortical function. So they sit in this interface between these inputs and outputs, which is somatostatin cells primarily, which are a major inhibitory neuron. And so they are sort of like a spider in the web, where they are in a good position to control cortical function. That was my hypothesis. So we started by removing ERBB4 in VIP cells, and that's what I'm going to call the mutants. So mutants is animals in which ERBB4 has been deleted in VIP cells, and then the controls are there just, control intermates in which ERBB4 has not been deleted. And we started by looking, using slice electrophysiology, to look at the miniature EPSCs into these VIP cells themselves. And what we saw is that when you look at the control in relation to the mutant, and control is always in black, mutant is always in blue, you see that there's no change in the amplitude, but there's an increase of the interval or reduced frequency of the mini EPSCs, therefore suggesting that there's a reduced glutamatergic input into VIP interneurons. Now, it is also known that the VIP interneurons receive not only glutamatergic, but also cholinergic input. So what we did in this case is that we puffed acetylcholine on top of the VIP interneurons, and we asked, are those neurons, mutant neurons, still able to respond to this puffed acetylcholine? And the answer is yes. So they can still respond to acetylcholine. Maybe they are not receiving acetylcholine inputs. I do not know that. But if they are presented with acetylcholine, they respond normally. Okay, so from now on, most of what I'm going to show you is actually experiments in which were performed in awake mice that are head-posted and are free to run in a wheel, has been trained for such. And then I'm going to show you either extracellular recordings from tetrads from in the visual cortex, from a variety of different cells. And I'm going to also show you calcium imaging to probe the activity of different populations of cells. So here I'm going to start to show you what is the calcium. So here we are doing looking at the calcium signals in a mouse that is on a wheel. And it has been shown, pioneer work by Michael Stryker, but followed by many others, that the VIP cells respond to locomotor signals. This means that VIP cells are mostly not active when an animal is sitting here. So this is the calcium traces delta alpha over alpha line to locomotion onset in the dotted line. And you can see that most cells, most VIP cells, are not responding when an animal is sitting. And the moment it starts walking, there's a robust response. And that has been shown by others before. In the mutant, this recruitment of VIP cells by locomotion is no longer present. So you can see that we don't see this nice, beautiful pattern in our mutant cells. So then we wanted to understand what happens when we do, alter the activity of VIP cells and alter the capacity to respond to locomotor activities. What happens to the main output of the cortex, the excitatory neurons? So what I'm going to show you now, it's by recordings when you look at the excitatory neurons. So to do that, we did the extracellular recordings of a mouse that is placed on a wheel. We monitored wheel position, pupil diameter, and other aspects that I'm not going to focus. And we looked at the local field potential that corresponds to the sum synaptic activity of the local network, as well as the activity of many individual neurons, here represented by spikes or action potentials. And here I just show a few, for example. And we can sort what kind of these neurons are in terms of waveforms. So most of them are excitatory neurons, or they are putative excitatory neurons. I'm going to treat them as excitatory neurons. And then we compare controls and mutants. And so if you look here, what is striking is that it looks like excitatory neurons in the mutants are firing much more. So we went ahead and we quantified this phenomena. And we quantified, in the adult, which I'm going to start to show. And you can see in adult mice, in fact, the firing rate of the mutants is much higher, about 4-fold to 5-fold to the ones of the controls. Now, I didn't tell you yet, but we are inducing this alteration of VIP cells around P7. And, the removal of ERBB4 is complete by around, mostly complete by around P20. So we went back and asked, when is that this firing rate is being altered? So you can see here at P15, even though most of the ERBB4 already has been removed, controls and mutants still show the same firing rate. But as time goes on, you can see that in the controls, actually, the firing rate reduces, which is something that also has been shown by others, but in the mutants, it increases. We were pretty stricken by this result, and we asked whether we could be able to rescue it locally by reinserting ERBB4 in the VIP neurons just of the visual cortex. Of course, the brain is all wired, is all together. So we were wondering, is this a product of something that is happening within the cortex, or something that is being altered in the thalamus or any other structure? structure. So we reinsert ERBB4 just in the visual cortex of the mouse, around at the same time that we are removing it, or soon after. So we are removing it at P7, we are inserting it around P10, and we saw that we could rescue the phenotype of firing rate at this age. We went then and tried to characterize the finer temporal patterns of activity in the visual cortex and ask, are those altered or not? And since those are highly linked to sensory function and cognition. And so here I'm showing spike trigger averages of two rhythms. So we take the local field potential and we subdivide it into the different components of oscillations that make part of it. For instance, here the slow oscillation, 1 to 6 hertz, or the gamma oscillation, 40 to 60 hertz. And then we basically do a PSTH between a continuous variable, which is the local field potential or the oscillation at a certain frequency, and a spike, right? One cell, individual cell. And it is known, it's been shown by many, that most cells sort of fire along with the rhythm, so they are called to be entrained. And so that's what you're seeing here. So in the control, in black, the cells are firing with a slow oscillation as well as with the gamma oscillation. In the mutant, that pattern was totally altered. So it's like, cells are firing more, but are like sort of not listening to what is going on around them. They are dissonant with the rest of the network. We also look at other form of synchrony that is in between cells. So we did basically cross correlations between two different cells and see how do they fire in relation to each other. And of course, cells that receive similar inputs should fire at the same time. So there should be a peak at 0. And you see it very well in controls. But in the mutants, that was also disturbed. It's again like the circuit is sort of like dissonant. So we know that cortical activity is highly influenced by behavior state, and the behavior performance is also very well dependent of the state of the animal. There's the beautiful work by David McCormick and others showing that depending on the arousal state of the animal performs better or worse, and it's highly dependent on that. And we were asking, and we all of a sudden see that not only the network is not in sync with itself, it's like, each cell is sort of a snowflake of its own right, but also there's no locomotor signals coming to VIP cells any longer. So we ask what happens to the flexible capacity of the brain to adjust to different stimuli or to adjust to different conditions, such as different behavior states. So here, what I'm showing you is an example of one cell that we are recording and that we are aligning to locomotion onset. So here the animal is not walking, here is walking. And you can say that there's this anticipatory response of excitatory cells to locomotion due to arousal. This has been characterized by us and many, many others in the field now. And so here's the population analysis. So this is a typical response. And in the mutant, that relationship is totally gone. So now the excitatory neurons no longer sense, as far as we can tell, signals of changes in behavior state. So this is the quantification, and you can see it's like a striking effect. All right, so we were like really puzzled. I must say that most of these results were actually the opposite of what I expecting, because if you reduce the activity in VIP interneurons, we expect to have actually not increase, but reduce the reduced activity of excitatory neurons, given the target is another inhibitory neuron. So we were, even a bit puzzle whether it's like, is there something else going on again? It's like some phenomena that is independent of the cortex that is sort of overriding what's going on, and why are we having these striking phenotypes? So I recently did this other experiment in which we manipulate the activity of VIP interneurons by a different method, by expressing KIR 2.1 inward ratifying potassium channel that basically makes the VIP cells much less silent, not totally silent, but much less active. And we did this age match at the same time when you remove ERBB4. We did it only in the visual cortex, so this manipulation is restricted to the visual cortex, right? Contrary to the other one that was loss of ERBB4 in all VIP cells. And you can see that you can recapitulate the most of the phenotypes that we have. So this is still preliminary. I think I have now n equals 5 animals, and I'm still working on this, but you can see that it's significant. So you can so when you make VIP cells less active during development. Postnatal development, we have still increasing firing rate and altered no transition states any longer. So we are doing all of these in the visual cortex, and obviously want to ask, the next step is like, what happens to visual responses in the cortex, in the visual cortex? So we now are going to do experiments in which we give a visual stimulation, a visual stimuli to the mice, which is a drifting grading of different orientations, just as you've seen before, that is being placed in the receptive field of where we are recording. And so this is some of the results. I'm not going to show everything, but anyway, so here we are looking at the signal-to-noise ratio of the population of excitatory neurons to drifting grading, right? And so you can see that there's obviously visual responses. It's the visual cortex, that is not surprising. But in the mutant, these visual responses are really reduced by a huge factor. We also looked in the receptive field properties, such as orientation selectivity and spatial frequency and some others. And here I'm showing one just for illustration purpose. So here we are looking at the orientation selectivity index that basically is the preference of given excitatory neuron to a bar of this orientation or different orientations. So you give a range of orientations, and then most cells prefer this one versus this one, and we call their preference orientation, their orientation selectivity. So you can see that by P15, where we didn't see any difference in firing rate, we don't see any difference in the properties such as orientation selectivity and others that we look at. But by P25 and in adulthood, there's a difference, and it's reduced and considerably reduced. So again, we tried to understand whether this phenomenon was driven by a property within the cortex or outside of it. So we did a rescue experiment in which we reinserted ERBB4 in VIP cells in mutants in which had been removed just a few days before, and we can totally rescue the visual effects. Now, I told you about these cells, the VIP cells. I told you about these cells, the excitatory neurons, and of course, your question is going to be, what about the one that is in between, that is sort of, the one that is mediating the effects, the relationship between these two? So that's what I'm going to tell you about right now. So we asked, what happens to somatostatin cells in these mutants? Now, to do this question, we had to use an intersection approach in which we remove ERBB4 with a CRE VIP line in VIP interneurons, and then we labeled somatostatin interneurons by using a flip allele, a flip line. And in this case, we use a virus that is flip dependent, so it will express the GFP or YFP in somatostatin cells in controls and mutants. And we then go and we measure the miniature iPSCs, inhibitory postsynaptic currents, in controls and mutants. And we saw that, in fact, in mutants, there is an increased interval or reduced frequency in the mini iPSCs of somatostatin cells, suggesting that there's reduced inhibition from VIP interneurons to somatostatin interneurons. So we then wanted to go and actually ask a little bit further and ask, how do these somatostatin interneurons act in the local network? And so we decided to do calcium imaging to probe the activity of somatostatin interneurons as a proxy of firing activity. And again, we did an intersection approach in which we are removing ERBB4 from VIP cells and looking specifically at somatostatin cells. And here I'm showing one experiment, an example experiment that has been replicated many times, in which we actually look at the activity of somatostatin interneurons relatively to locomotion onset. And I showed you before that these cells are controlled by, respond to locomotor signals in the control, and in the mutant that is disrupted. And here what you see is the opposite in the somatostatin cells. So in controls, most of these somatostatin cells are really not responding to locomotor signals, but in the mutants they start to. And so which was, again, another rather surprising result. We then ask, how do these somatostatin cells respond to visual stimuli in control and mutants? And so what we saw is that there's a huge reduction in the visual responses of somatostatin neurons in the visual cortex when the mice are presented with the drifting gradings. Okay, so as we just heard recently, nothing in biology or in neuroscience makes sense if not in light of behavior, right? So we wanted to go all the way from a mutation in a specific gene in a specific cell type to alterations in the patterns of activity within the cortex to changes in behavior. And so we designed a visual detection task very much in line to what we just were shown recently, where animals are trained in a go, no-go task. They are shaped over a few days, a few weeks, in fact, in this task. And eventually what they have to do is to detect drifting grading of changed intensity, so from high intensity to low intensity. And if they get the task, they leak and they have a water reward. And if they don't, if they leak when they shouldn't, they get a timeout. And so this is the first time we got this task to work, so I got very excited about making a video. And this is mainly a video to show for the ones that are skeptical that mice can see. Here is the stimuli, and you can see that he could very reliably detect that very low contrast stimuli. Actually, in my first trials and the mice first trials, we were as good. I was just much better at improving very quickly, where the mouse took much longer to improve. But the first times, he would get it as wrong as I would, first times I tried. So mice can see, and actually they appear to be seeing quite well. So then we compared the psychophysics curves between mice, within controls, in mice when they are just first learning the task. So this is here what I'm showing you in the dotted line is the first 2 days when the mouse is being presented with this task of reduced contrast, right? And you can see that they are very good in these early mice or these naive mice. They are really good at high contrast, but they really are not so good at detecting low contrast. As time goes by and these mice become experts, and here you have a curve of the expert mice later on, they are not just good at the high contrast, they are much better at the lower contrast still. There's a threshold that they can no longer detect. So we did a similar analysis for mutant mice, and if you can see is that the early or the naive mice, they are not so different from the controls, but they never improve, suggesting that their threshold for detection is lower, or they have learning deficits. We cannot really tell this apart right now. So here's the quantification. So it's in early there's not significant difference, but late there's a massive significant difference on the performance of these mice. Okay, so I am gonna now summarize with what I told you. So we did a manipulation in these VIP cells in which we reduced their activity, here represented by reduced glutamatergic input. And when we look at these cells during development, and that is an important factor, when we look at these cells after we reduce their activity in the adult brain, we saw that they no longer are responsive to locomotor signals. Furthermore, once there's reduced input into somatostatin interneurons as assessed by mini iPSCs, these somatostatin interneurons now have a different relationship in terms of how they respond to both locomotion and visual responses, and that eventually leads to profound alterations in the excitatory circuit, both in terms of really drastic reduced capacity of entrainment and synchrony and alteration in the fine temporal patterns of activity, as well as inability to respond to changes in behavior state, such as arousal state, and alterations in both visual responses and visual perception. Now, let me go back here one more second. So now I'm going to tell you what I'm going to do in the next couple of years. I'm just about to start my own lab, so this is exciting times, having all this data that is like a lot to interpret and a lot to dissect further on. There's like many open questions that we have to go after. And what I'm really interested is actually about not just the mature function of these neurons, but how they become to be who they are. So we heard from Christoph that it will take about 100 times to understand how all the different interneuron types or neuronal types, not just not just the way that they are represented here, but the way that they were represented yesterday by Ed Schematic to understand it all in the mature brain. But we already know a fair amount of how some of these neuron types interact in the mature cortex, specifically in the visual cortex, that a lot of people have been looking, and how their function influence both sensory perception and behavior. What we really do not know is that how they come to be like that, right? So the local network has to go from birth, from a state that is basically zero connectivity, to a period that is massive connectivity, and there's like the cells are just like talking to each other, creating some oscillations, figuring out what's going on, to a period where sensory input comes in the mouse, like around 15 days of age, and now like there's sensory information coming and pruning the responses in the local network, and evolves through a series of discrete and continuous landmarks during development to the precise circuit able of rendering such sophisticated functions as it is. And we do not know, we know a little, we know quite a great deal about embryonic development. What are the cell types? Where do they arise from? How do they migrate and get into the cortex? And then we know, now once we have the cell types here, they interact in these specific ways. They target these different areas. And, what is in between is really a black box. And that's what I want to, contribute towards understanding. So my research program is going to focus in the postnatal development from day 0 all the way to the adult. And it's going to ask a few questions. It's going to first start by observing, actually. So how does the patterns of activity evolve during development? Which cell types come on board first? Are those the ones that are going to then regulate the activity of other ones? Which are the cell types that are involved? Then are going to ask, how do we, if we manipulate these cell types during development, how do we change the patterns of activity during development itself? And then, final on, if we manipulate these cell types in sort of both with optogenetics or with other molecular tools, how are we going to influence not just how the brain processes information during development, but how it's going to process information in the mature cortex? And I think that is going to be exciting, and we are going to learn a lot about it since not much is known, and it's going to be important from the perspective of basic science and from the perspective of more applied science, since that most of the neurodevelopmental disorders, such as schizophrenia and autism, arise during these early ages. If you do intervention during these early ages, can be corrected to a certain degree because the system is plastic enough, and so I'm going to do it not just looking at optogenetics, but I'm also going to do it within the context of removing specific genes that have been linked to diseases and reintroducing them at specific ages. So if you are interested in these questions, I am recruiting. So that's, I just need to thank very briefly to everybody that has been involved in this project. So first of all, Jess, I did all this work that I show you in her lab, and I'm transitioning now in my own, and she has been a great source of inspiration of how, to become a young PI. Martin helped a lot with some of the heavy lifting of the data analysis, Katie with the 2-photon, and Michael Higley with the slice electrophysiology. Thank you.
All right, we have time for just a couple questions. Someone will find you with the microphone.
Yeah, I just wanted to ask you if you've done any experiments. I mean, presumably the schizophrenia phenotypes, for example, reflect differences in activity, not absolute elimination of, for example, ERB for activity. So if you looked at, for example, heterozygous deletions or impact of overexpression, for example, in the control case?
So I did look a little bit of heterozygous, and it seems to be like a somehow in between phenotype. So those I think matters. I also looked actually, and this is a paper that we are trying to wrap up, what is the function of the same gene RBB4 in a different cell type, provalbumin cells, and the effects are different. So we, the same gene is going to have different impact in if you remove it in VIP cells versus provalbumin cells. So, it's.
Hi, I wanted to know if you had any information about how the removal of ERB4 affected inputs of inhibitory cells onto the VIP population?
So that's where we are, one of the things that we are trying to do right now. So we are looking actually into both inputs and outputs from these cells, and I suspect that it's going to be radically changed, and that's why we are seeing such striking phenomena, but we don't know yet.
All right, I don't want to keep you from your coffee much longer, but if you have more questions for Renata, please track her down during the coffee time. Poster presenters, if you've not yet put your poster up, please do so during the coffee break. And everyone, please join us back here at 10:50 for the third year Next Gen Leader panel. Thank you.